Okay, so I'm going to talk about what we've been working on in the space between computational biology, clouds, and sort of data analysis and pipelines. So these are full pictures. These are platforms, which we wish we were working on. But we are working on this kind of stuff. So these are a couple of illustrations of what it might be like if you could look at a cell, in this case E. coli, at 1 million x magnification. So, of course, at that scale, we can do electron microscopy, but we don't get colors and we don't get any kind of 3D rendering of this. But the point of this, these pictures was to show that even in the simplest kind of bacterial cell, that the complexity is quite high. The interior of these cells isn't like we might imagine from when we were in school, where we were, sort of the naive notion was that cytoplasm inside a cell is like a bag of water with a little bit of stuff in it. In fact, the inside of bacterial cells is very tightly packed. I mean, there's water in here, not illustrated in these pictures, but there's a lot of protein molecules, ribosomes, there's very complex membranes, aren't smooth, they're decorated with all kinds of surface proteins and lipids.
So Rick, it's a human cell? Yeah.
Sorry, this is a bacterial cell.
Is a human cell similarly complex?
Human cell would be even more complex. And I used to have some cool movies from Harvard that would be like, zooming around inside the endoplasmic reticulum and stuff. But yeah, I mean, cells are really complex complex objects. Bacterial cells are cut maybe 10 microns, 2 to 10 microns long, a couple microns wide. And you have molecular machines like the flagellum sitting here that has about 40 genes that make this thing up. You got some DNA coiled around. And of course, what we're trying to do is understand this not at the level of individual proteins as much as at the level of the system. This is the idea of systems biology. And of course, these organisms interact with their environment. Here's an example of a channel through the membrane that can traffic certain compounds. So this is just to give you some kind of a gestalt of the ideas that we're working on. Of course, behind all of this is you've all learned as sort of the central dogma in biology. So inside the cell, we've got an information system that's based on DNA. We've got machinery that transcribes off that DNA. Then we translate these RNAs into proteins. Some of that protein is used for maintaining DNA. Some of it's used to drive this set of processes here. A lot of it is used to build biochemical circuits that then enable the cell to actually function. And of course, what we're interested in when we ask kind of high-level questions about biological systems, and I suspect Gary will talk about some of this, is the relationship between what's in the DNA and what is expressed, that is the phenotype, that is what the organism actually doing under some environmental conditions, right? And, it took biology on the order of 100 years to sort of work this system out. And now we're trying to, of course, collect information on many, many thousands, tens of thousands, hundreds of thousands of organisms by sequencing the DNA. And then for a set of model organisms, interrogating that system with different technologies that allow us to snapshot what's happening in each of these stages. So we can use DNA sequencing to read the DNA. We can use RNA-seq or gene chips to snapshot what's happening at the RNA level. We can use proteomics to snapshot the state of the cell for proteins. We can use metabolomics, like mass specs or chromatography, to get an inventory of the small molecules that traffic in here. And of course, we can do all kinds of phenotype measurements, like what does this cell grow on? What does it look like? If I challenge it with something, how does the cell respond, and so forth. So we can collect information all along this side. And from this collection of information, it's often called omics, this idea of adding omics to some types. So we have metabolomics down here, and proteomics, and transcriptomics, and genomics here. We get enough information that we can recover the state of the cell. This sort of big inverse problem, reconstruct what's actually happening in the system, and then model that as a system problem. And of course, if we do that for a single genome, get to the pathways, and model it as a single cell, we're also very interested in going from a single cell, single organism, single kind of effect organism, to a population of cells, to a more complex community that might be in a lab, microcosms, the Winogretsky column, where we grow bacteria inside a column with cellulose, and there's like oxygen at the top and no oxygen at the bottom. And the different species of bacteria will align themselves in the column based on the kind of metabolism that's going on in there. You can interrogate this. It's been a really clever thing for 100 years. But then in real environments where you might have thousands or tens of thousands of organisms interacting. And so we ask different questions. Here we're trying to reconstruct a single cell or a single set of biochemical networks. Here, a whole organism. Here, organisms with variation and how they interact with their peers. Here, where they interact with other members of the species, and here where they might interact with a really complex environment. And we have similar techniques that I showed on the first slide for interrogating each of these different levels. But here you're asking sort of ecological questions, and here you're asking questions about individual genes or proteins. Right? Now, in order to convince ourselves that we understand these systems, what we ideally want to do is build models at the different levels of abstraction, and then make predictions and then test those predictions. And one way to think about building models in these systems is the kinds of models and the relationships that we might build. So if we're trying to think about a single protein, say, or a set of proteins that might have a simple kind of interaction, we might have very mechanistic models where we're trying to model the actual physics or the biophysics. If we're trying to model, say, the input and output relationships of, say, some pathways, we might have a functional model that doesn't try to reproduce the detailed physics, but tries to give us some idea of the function of that system, right? And then if we're looking at systems where there might be abstract kind of properties going on there, for example, in macroscopic ecology, you have predator-prey relationships, right? Wolves eat moose and stuff like that. In microecology, we might have protozoan eating bacteria, right? Well, we can describe those relationships using the same kinds of models, predator-prey systems. In fact, we can even take that kind of a predator-prey model and use it to describe relationships between molecular entities inside the cell as well. There's cells that, or molecular entities that sort of bind to and immobilize other cells. We can treat those as predator-prey relationships. So we go from very detailed mechanistic models, which are really not even biology at all, this is physics and chemistry, to things that are really biological concepts, to things that are more like generic theoretical concepts at higher order entities. And the combined set of this would be a set of, collection of conceptual models that implement sort of a theory of biology. And what we're ultimately interested in doing is trying to push this theory to the point where we have the ability to say something useful about the world. Okay, so and do this not just for individual molecules, but all those levels that I was talking about in the heart. Now, there's another thread to this, right? So, how many of you sort of started out your academic life, as a 10-year-old or whatever, thinking about physics or math as opposed to biology or computers? Okay, all right. So, one of the ideas that you probably encountered along that path was this idea. Leibniz had this idea, Newton sort of had this idea, Leibniz was really the first one to sort of articulate it. But if we could write down rules, right, we think in terms of the calculus, but or generally about the physical world, if we could capture things in rules, then we could calculate what was true by just applying those rules and logical rules of inference, right? And that, the philosophers, of course, had this idea too in logic. And in the 1920s, there was a huge interest in applying that methodology to many areas of science, right? Russell tried to apply set theory. Lots of people in the positivist movement in Austria or in Eastern Europe started applying this to different areas of mathematics and logic. Ultimately, of course, they ran into some trouble with the incompleteness theorem, but But it spawned this idea that if we can encode science as a bunch of rules, that we can compute what's true, right? And then we, of course, go out and look at something in the world. We can compare what we see in the world to what we can compute, right? And that concept worked pretty well in the foundations of mathematics, and it works sort of in quantum mechanics. Around the 1930s, this guy, John Woodger, attempted to do this in biology. And what I've reproduced here is a couple of books he wrote. When he tried writing down, at that time, he tried to axiomatize and write rules for what was true in biology. And of course, he spent years, decades on this. We didn't have computers then that could do anything with these formulas, so the ability to calculate was a hand manual process, right, using inference rules. And this was an utter failure, okay? And it was an utter failure because in the 1930s, we didn't understand how this worked at all, right? We had really primitive notions about how biological systems work. And so what we encoded was not true. It was these very high-level kind of phenomenological things, which didn't give you any access to truth. So this was a great, I mean, I'm really inspired by this, and I'll talk more about it in a minute, but it was an utter failure, right? And it was because this underlying science wasn't ready for this kind of encoding. Fast forward, I don't know, 80 years, right? And where are we now? Well, we're trying to push this agenda, that is this idea of building theory in biology. But we realize that biology is really complex, right? And even the simplest system, right, those bacteria pictures I showed you, is incredibly complex, right? 4,000 genes in E. coli, millions of actual instances of molecules. So you can't really just reason about that as a simple system. It's very data intensive. So how do we proceed on this agenda? Well, what we think we need to do is we collect a lot of observations, hypotheses, and experimental data. So we collect both what we've measured and what we hypothesized. We organize this, right, in some clever way. We devise means to search it, query it, analyze it, compare, add nodes, annotate, update, and so on. We build tools to allow us to extract models from these data collections, right? And be able to then run those models to make predictions, and compare those to what we're measuring in the world. Update, the parts of this that are our rules, basically. And then synthesize principles from this loop and sort of crank on this, turn the crank on this a lot, maybe for 100 years or 1,000 years, I don't know how long it's going to take. And do all of, realizing that we have to do all of this in some environment that's fundamentally enabling of collaboration. Because first of all, biology is a huge scientific area. It has more people work in biology than all the other sciences combined, essentially, except for chemistry. No one person can sort of get their head around the system, so you've got to be able to collaborate. So that's this is what we're about, right? Now, if we look at, I'll walk you through some of this, and I'm trying to go fast, and Ian should stand up if I run out of time. But you say, well, how big is the universe that we're trying to integrate over? Well, back in 2001, when we started in earnest on this, there were about 300 biological databases. In 2011, the last time I computed it, there was over 1,000 databases, right? So not thousands of data items, thousands of databases, each of which could contain thousands or millions of data items. So huge amount of data, right? Massive numbers of databases. What the community's been doing over the last decade or so is building tools. Typically, here's a half a dozen of them, some we built, some of our friends have built, that are web-based tools for some subdomain of analysis. So, for example, Phytosome here. This is a tool built out of University of California and JTI for analyzing plant genomes. Okay, it's got, this is a couple-year-old slide, but 25 plant genomes and 300 people poking at it every day, sort of doing stuff on it, right? We have one for metagenomes, that is samples from the environment. We have about 40,000 metagenomes. 500 people a day are adding data and doing analysis here. We have a system that annotates microbial genomes, that goes from the DNA sequence to function for like E. coli, like we were talking about. And this is really pretty old. We have now about 160,000 genomes in there and about 14,000 users, and so forth. So there's people who are building systems for specific parts of this problem. But what we discovered is that, and making data open and so on was great, but it's not really sufficient to do what we want. And the reason for this is that how many of you have a personal petabyte of storage in your, I don't know, apartment or dorm room? How many of you have a 10,000 node cluster to analyze that petabyte data in your room? Okay. So the fact that all the data is open and in principle touchable, that is, you can go out on some URL or URI and grab a piece of data, is absolutely no use to you, right? That is, if I tell you, oh, there's this petabyte of data you got to analyze, you're stuck. So we've got to somehow figure out how to democratize this analysis, right? So the data can be wide open, but individual users just don't have, everyone doesn't have access to this stuff, right? The data infrastructure limits, you've got constraints on what you can compute, and maybe more critically, these can just be solved with money. Trying to build an environment that has hundreds of tools integrated in some useful way that you would need to make sense out of that is actually really hard, right? I mean, not just because every tool has some special knowledge to make it work or some expertise to make it work, and integration is, a mess. So what we're trying to do is sort of build a system that sort of, democratizes stuff and tries to address these to the degree we can, so we make this easier for people to do, right? And what do we want people to be able to do? Well, we want them to be able to explore the data, do integration, to integrate data, different kinds of data, to form hypotheses in this context, to do inferencing on it, or build models, test the models, right? Run a lot of tests, maybe discover something, and then tell people about it, right? So this is what the environment sort of has to be able to do. And one of the other problems, for those of you who are like hardcore CS people, algorithm people, as the data gets larger, we have to keep shifting gears on the methods, right? So back in the 60s, when we had all of about 1,000 protein sequences, you could use an order n squared algorithm for comparing those sequences. We had slower computers, but we had just really tiny amounts of data, right? So you could do a sequence comparison in a very slow kind of order n squared algorithm. As we started to get more and more sequences, people said, well, we can't really do that, it's too long. We still have the algorithms that were fine, they've just been practical. We started building algorithms that use heuristics or that have some better time complexity. This is many of you probably heard of the idea of BLAST, this basic local alignment algorithm that has heuristics in it, takes a problem that would otherwise be sort of n squared and makes it order n log n. And now data sets are too large to even BLAST, and so we're trying to use methods that are maybe linear or sublinear, right? So the algorithms have to keep moving to keep up with the class of data. And I already talked about some of this stuff. Let me zoom forward here. So one of the projects that I'm involved in, or my group is involved in, is called KBase. And KBase stands for Knowledge Base. It's a short for DOE Systems Biology Knowledge Base. So it's the project to build this thing that I was describing. Okay, both the data, the methods, the tools, the models, all in an integrated environment. And we're trying to build this thing to support predictive systems biology, so we can say something about the behavior of the biological system, not just a single entity in the system, and make it in a way that people can extend it, right? And it's all open source, and it's all available in some sense for people to grab and build on. And it sits on a real infrastructure, and we're trying to standardize the kind of internal components in a way that makes this work. And we're focusing on 3 areas. Microbes, like I was talking about before, microbial communities. Remember that chart that showed the one genome down here up and over to these complex communities? Well, one is focusing on individual bugs, and the other is focusing on these complex communities. So that's the second part. The third area is plants. And the reason that these 3 are chosen is because the Department of Energy cedes all the biomedical work to NIH, right? So NIH, if they were building a version of this, would have humans and microbes and protozoan or something in there, malaria organisms, right? So KBase is both a platform that has the abstractions and models, user interfaces, APIs, a bunch of services, and data stores in it, but it's also actually an infrastructure. So we have a a bunch of cloud compute servers, a bunch of data servers. It's replicated across a couple of different sites. We have 3 different database technologies: relational databases, NoSQL databases, object stores in there. There's a lot of interest like networking. It's security infrastructure, and it's extensible in a cloud sense. So when we have services that need to scale up, they can spill over into more elastic services. It's built on a service-oriented architecture, so what users see are endpoints of services that can scale sort of independently from each other. And so one day, if there's a lot of people doing a certain type of function, like assembling genomes, and the next day they're doing modeling, we don't have to scale the system up in some kind of linear way. We can expand resources for specific services. And it's also client server, so the services are implemented in this split fashion. And so when we were adding things to this, we set up and deployed new services. And each service might have a bunch of internal complexity to it. Right now there's about 1,000 functions in this thing, maybe 100 services. And I already mentioned that. Right? And roughly it looks something like this. I mean, there are these different database types depending on the type of data. So we have relational databases for highly structured data. We have what we call an auxiliary store or bulk store for really, really large things like data sets that might be terabyte sized. You don't want to shove those in a relational database. You want to shove up the metadata in the relational database and the big data in a big file system. And then workspaces which support sharing and privacy and so forth. And then generalized access and search layer, and many, many pipelines that are back-ended on the cloud, and then this kind of dispatch, and then various kinds of interfaces. So you can use it from a command line, from a remote invocation service, or this kind of very like Python-y kind of thing, and various web pages and so on. So this is sort of the general concept of how it's put together. Now, what kinds of things can we do with it? Well, for microbes, right now, we have all these kinds of features in there. So we can start with the raw data that comes out sequencing machine. We can assemble that data. We can annotate it, predict features. We can do comparative analysis across hundreds or thousands of genomes for certain things. Do evolutionary analysis. We can then build models. So we have lots of pipelines. Each one of these stages would be one or more functions in a service. This entire pipeline is spanned by about 3 or 4 services. Give you some sense over here. Sorry for going so fast here. Once you pop something out of that, you can then do modeling with it. We built about 100,000 models. Some of these models are used, for example, for predicting drug targets. So one of the uses of this pipeline is to sequence a bunch of pathogens, annotate the organisms, build a metabolic model of those organisms, and then do a computational knockout where we systematically go through and disable the function of each gene and find a set of genes that, if they were knocked out, say inhibited by a drug, would make the organism not viable. This would kill the organism, right? So a couple years ago we did this, and we generated about, across a bunch of different pathogens, about 300 genes, or proteins really, that were deemed by the modeling to be essential in a range of pathogens that we didn't know the structures for. And therefore they couldn't be used for drug development, because you have to have the structure for doing drug development. We sent those over to our collaborators at Northwestern, who run the Structural Genomics and Infectious Disease Center, and they tried to get the structures, and about 18 months later, we got 23 new protein structures in the protein database based on the modeling. Okay, so the entire input to that prioritization was coming from our predictions of what was going to be essential. And then we can do things like figuring out, which genes are more likely to be expressed under environments, trying to fill in holes in metabolic pathways by doing inferencing and modeling together, and rank candidates as possibilities for missing genes. We can do evolutionary studies, right? We can also apply this to metagenomics. Don't forget this stuff. So we have a similar pipeline now that's much higher performance and uses different technology, because the typical data set we're dealing with here, instead of being a single, say, 4 megabase pair genome for a microbe, might be a couple 100 gigabyte data set that comes from sequencing an environmental sample, but we want to try to figure out where each of those reads came from in it. We want to assemble it to the degree we can, recover genes or recover collections of genes, predict function, try to make some sense out of what's happening in that environment. Okay, go with things with it. So we're sort of headed toward this direction where, we have integrated biological data, we have this loop with bioinformatics tools, we have the ability to sort of plan experiments, do the experiment, integrate the data, and sort of go on this loop here, but also do this loop where we do analysis of the data, plan computational experiments, run the models, sometimes at large scale, make a set of predictions, and ideally if we're doing the same thing with the experiment at the same time, then we reconcile the experiment with the simulation, trying to correct our underlying, say, notion of what the function should be. Now, where are we headed with this? What we ultimately want, right, is this. What we ultimately want is a set of rules, right? And maybe what we discovered is that, unlike physics, where physics is very data light, and we can describe, most of physics with a very small amount of data or parameters in our models, biology maybe theory is really going to be data driven, so we have big statistical models underlying things because of the complexity or the variation. But we want to move to something like this, where we have a system where we can sort of query it, and it will tell us what we know about something, a combination of looking up the data or computing it, right? Now, the best systems today that do stuff like this are sort of question answering systems. So this is one from MIT, this is called START. You should play with it, it's been online for a bunch of years actually. And they've ingested a lot of databases, mostly in things like, space science, geography, sort of Watson-like, and you can ask it questions. You can say like, what's the tallest mountain in California? It'll come back and tell you, Mount Whitney, blah, 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 right? We want the equivalent of this for biology, where we can say, what are the 3 most common genes that cause this abnormal phenotype in some organism? And the system would actually be able to answer that question, or give us some ranking, right? Wolfram guys are building something similar, right? Wolfram Alpha, which is their attempt at making all kinds of data computable, right? And so this morning I was putting this together and I said, well, what is it? I asked you really, actually you got to do this, you always got a machine. Go to Wolfram Alpha and ask it, what genes cause cancer? And of course, it doesn't know about cancer as we think of cancer. It knows about cancer as the constellation Cancer. And it doesn't figure out, it doesn't know what gene is, right? And so it brings up all this information about the constellation Cancer and then all the historical information about that, right? So it'll tell you how hard this is. But if you type in something like Staphylococcus, which is an infectious organism, it knows a little bit about it, right? It sort of brings up some stuff, maybe a little bit more stylized than Wikipedia. But what we're after is a system that we can sort of discourse with that has all that machinery I'm talking about behind the scenes, right? That's constantly reading the literature, it's getting the new data, it's integrating, it's running these pipelines, it's making predictions, and what's made available to the end user in some sense is a very clean way of really querying and interacting with the system. So anyway, that's my talk, and I'm done. That's great.
Thank you.